So, chapter 5 is about linkage, recombination, and eukaryotic gene mapping. And we're going to start with some fundamental review of a couple of concepts, and then we're going to lay out some rules and learn how to create what we call genetic maps. So there's some problem solving in this chapter, and you'll need to practice. So we have a lot of practice problems that go with this, and you will be able to master this kind of problem. So let's just start with some of the background information. You may remember that linkage refers to a condition in which either a gene is linked to a chromosome. So we've talked about a little bit about X linkage, when genes are located on the X chromosome, or in this particular chapter, we're going to be talking about linkage when two genes are linked to each other. That means they're located near to each other on the same chromosome. And so when genes are linked to each other, that means they'll be inherited, they tend to be inherited together rather than independently assorted. And really, we mean when alleles are linked to each other, they will tend to be inherited together. But I'll be explaining more about that in a minute. What we're going to be doing, though, is measuring or calculating the extent of the linkage, meaning how close together are two genes located on a chromosome, so that we can construct what we call genetic maps. And that's a particular kind of map. Any kind of map depicts the linear order of something, in this case genes, on a chromosome, and then the relative distance between the genes. So if you were creating a map, let's say, of a highway and then the cities along the highway, for example, in Dallas, if you come from the south on 75, if you're going towards the north, you would pass through Richardson and then Plano and then Allen. And so the map would include Richardson, then Plano in the center, and then Allen on that route, and then the relative distances between the cities. So in a similar way, we're going to be creating genetic maps where we have the order of genes on a chromosome and then a measurement of distance indicated between the genes. Now, when we looked at Mendel's crosses, he only dealt with non-linked genes or unlinked genes. So you remember that when Mendel did his dihybrid cross, and you have, right here, you have the dihybrid individual creates a potential of 4 genotypes of gametes. If we're using gene A and gene B, then the dihybrid would produce the big A, big B gametes, the little a, little b gametes, and then big A, little b, and the little a, big B gametes. And then the other parent would also produce the same. And all 4 gamete types would be equally likely. And so when we put them on the Punnett square, when we put all 4 types of gametes across the top from the types of eggs, for example, and when we put all 4 gamete types down the side for the types of sperm, we're indicating that they're equally likely to exist in that parent and to be able to fertilize another gamete. And that's why we end up with, you'll recall, a 9:3 to 3:1 ratio, phenotypic ratio for the offspring of the dihybrid cross. And we could treat the dihybrid cross as if it were 2 independent monohybrid crosses occurring simultaneously. So you may recall the math that's associated with that. For example, when you do a dihybrid cross, your offspring, you would get 9 out of 16 would be dominant for each of the traits. And that's because if we just look at this big A blank outcome, if we're just looking at a monohybrid cross, there's a 3/4 chance of getting big A dash. And there's a 3/4 chance if we just did a monohybrid cross for the gene B, we would get a 3/4 chance. And so we just treat them as if it's 2 independent events occurring simultaneously. And that's why that math works. But in this chapter, we're going to now address what if the genes A and B are not independently assorted? What if the genes are linked? So what I'm saying is, what if on one chromosome you have, for example, big A and big B, and on the homologous chromosome you have little a and little b? If they're close enough together, then when segregation occurs in meiosis, you'll get mostly gametes that have a big A with a big B, and or gametes with a little a and a little b. And so you'll get mostly those combinations. You can get the other possibilities. So the way I drew this here, if this was the parental cell beginning meiosis, then most of your gametes from that, at the end of meiosis, would have a big A with a big B chromosome or little a with a little b. You could get in some meioses, you could have a crossover that occurs, but that's going to be less likely. And so we say that most of your gametes will be these two types that I just drew, and then a small number of gametes will be these crossover types. So big A with little b and little a with big B. All right. So all I can tell you is when genes are linked, if you do a dihybrid cross, since the four types of gametes are not going to be equally represented, you will not get offspring in a 9:3 to 3:1 ratio. I can't tell you what the ratio will be because that depends on how tightly linked the genes are. But I can tell you that when you do a dihybrid cross, you won't get 9:3 to 3:1. And in this picture here on the screen, which I kind of drew on top of, but I think you can still see it, you have a dihybrid cross, right? You have the heterozygous purple flowers, long pollen, self-fertilized, so that's the dihybrid cross. And then what you can see down here is these are the four groups of offspring, but you can see, I think, that when you simplify these numbers into the empirical ratio, the simplest ratio, you will not get a 9:3 to 3:1. And so when this happens, when a dihybrid cross does not yield a 9:3 to 3:1 ratio, then what we would hypothesize is that the genes might be linked. And so that would lead us to then test for linkage. So the concept here is you've performed a dihybrid cross, but you don't get a 9:3 to 3:1 ratio, so you might decide to see if the two genes are linked. When genes are linked, they tend to be assorted together into the gametes. So like I said, in this case, the big A, big B, and the little a, little b would be called parental gametes or gametes with parental allele combinations. The gametes that have crossover chromosomes are non-parental, or you could say crossover allele combinations. So like I said, if you did a dihybrid cross and you didn't get a 9:3 to 3:1 ratio, then you might back up and take one dihybrid individual and do what's we call a test cross. And that would be you would take your double heterozygous individual and cross it to a completely recessive one. And this is how we're going to be able to figure out if the genes are linked and really determine the distance between the genes. We're going to use data to measure the distance between the genes. So linkage and what we would call the extent of linkage is revealed through a test cross between a double heterozygote, so dihybrid, and an individual that's completely recessive. So we will pick this up and continue in the next lecture segment.